Well,
Those are the
so
triggers.
no.
Thank you. Yeah, so thank you, and thank you for the invitation. Thank all the organizers for putting this together. It's my first ISOL conference, and I'm really excited to, I mean, I guess be with air quotes, but be here and being able to see the talks by the other presenters and participate. So you can see behind me and also in the background of my slide, these structures that I want to talk about today. This is a fancier version than the ones I'll talk about in more detail today, but they're coacervates, so they're liquid droplets filled with polymers and water, and they're in an aqueous background that's more dilute in their contents. And they can have multiple phases like these beautiful ones do, but they don't always, and it's a lot easier sometimes to do quantitative work if there's just one of the polymer dense phases. So that'll be the kinds of systems I'm focused on today. Let's see if I can get this to go forward. Yeah, so this community actually is probably more used to the word coacervate than most, because of Professor Alexander Oparin back in the day had thought that, hey, these structures, which had been under study by his contemporaries, people like Bungenberg de Jong, who coined the term, and he thought, oh, those, look like they could be protocells, and we can put molecules in them, and those molecules can be performing reactions, and so maybe that's like how life started. And it's true that since then, people discovered things like DNA and RNA and lipid self-assembly. And so attention kind of went away from coacervates. There were these other really important things that were clearly part of the origins of life discussion. But I want to sort of bring us back around to, we have new methods and new ways to think about these now. And I think it's time to revisit these as a part of that process. So what they're really good at is being compartments. And they can be combined with a lot of the other things that we've heard about to aid in compartmentalization. So coming back to what they are again, so the term really comes from the fact that this is an associative phase separation, where the molecules are basically sticking together, and they're sticking together weakly and dynamically. That's why you get a liquid phase instead of solids. You can get solids, but they're just less fun generally, and so we don't like to talk about them. But it's all sort of on a continuum, right? Depending on how sticky and how transient. You absolutely can have nothing happen, everything stays in solution, or you can have phase separation, or you can have aggregates formed. But we're going to focus on the part that makes these liquid droplet phases. And the particular kind that I want to talk about today are complex coacervates. And these complex coacervates are called that because what's happening is ion pairing. That's the type of association, and it leads to complex formation. And so the droplets that you get, we call them complex coacervates. They're not any more complicated than the other kinds. Basically anything that makes the polymers stick together transiently can make coacervates. Usually it's not as pure as just one kind of interaction, but we're going to pretend it is, and we're going to use simple model systems to make that assumption as true as it can be, just to make our lives easier as a starting point. And the idea is you can always sort of add complexity once you have some kind of foothold on understanding what's going on. So these kinds of structures have phase diagrams that look kind of like this. I don't know if I told you on the first slide, but I'm a physical chemist. I love a phase diagram. And what I want you to notice about it, so down in here, this is compositions and situations where two phases coexist, so where you have droplets. And if you come outside this curve, you just have a one phase solution. And so the things that matter for that you can see on the phase diagram, concentration of the polymers, we're going to assume that they're charge matched at 1-to-1 ratio here because it makes the phase diagram easier to draw. We're going to keep it that way in all of our experiments today too, so you don't have to think about it either. And then there's some kind of interaction strength variation here, and I'm going to be using increasing salt concentration, which is going to destabilize these interactions because they are ion pairing. So if it's salty enough, they will eventually go into solution and you won't have phases anymore. If it's not salty enough, they might be solids. And so somewhere in between is the point where you're going to have nice liquids. This is a microscope image of a labeled, I think it's a labeled RNA that's red, that's inside these peptide RNA droplets. What they're really good at, like I mentioned, is accumulating molecules that might otherwise be dilute and concentrating them into the droplet. So one thing that's nice about ion pairing as a mechanism here for the complex coacervates is that in the prebiotic chemistry community, we like RNA a lot and RNA-like molecules. And basically those are polyanions. And so we can really think very intuitively about, oh, which kind of coacervate is going to take my polyanion up really well? Like, oh, well, I need to have really long, really sticky polycations. Or how do I make my RNA less likely to unfold. Well, I need to make those polycations less sticky. And so it's pretty easy to intuit sort of how you can optimize the behavior of these and to interpret the results, which again, it is an oversimplification that's intentional right now. And I think, as we learn more, we can start to bring in more of the complexity. So we've done a lot of work with RNA actually in these systems. It's our favorite molecule to compartmentalize. And all of this work has been done in collaboration with Phil Bevilacqua in his lab. And I've mentioned Dr. Raghav Pudyal here, who did a lot of work that sort of listing some of the publications down there. And the upshot of this is that you can encapsulate RNA really well. It can retain its secondary structure. It doesn't always, but you can arrange for it to retain its secondary structure. And you can arrange for it to retain or even to enhance its catalytic activity. It does depend on everything. And I could give a long talk about that, but it's not what I want to do today because that stuff's already been out for a little while. And what I want to focus on instead is sort of, let's take my word for it that, yeah, it might be nice to use these kinds of compartments, to think about them as compartmentalizing early reactions and early molecules that might be useful. And if we assume that that could be a useful thing, then let's try to move towards something closer to the prebiotic environment. So when we've done a lot of these experiments, you come on a complex coacervate as a physical chemist and you think, well, I just want one that's well studied. I don't want to have to reinvent the wheel. And so we would use molecules that wouldn't be very prebiotically relevant, really, really long molecules, really, really cheap molecules, really, really simple molecules. And we would do it under conditions that wouldn't necessarily be prebiotically relevant. And so we're trying to take sort of baby steps towards getting closer and closer to relevance. And some of those steps I want to just quickly touch on today. You'll see that we're not there yet. A lot of big steps remain to be taken, but we're trying to get there. And one thing that can be really helpful to us when we talk to people who've been thinking longer than we have about prebiotic chemistries, if they could sort of narrow down for us, like, oh, these kinds of experiments are the really important ones that you should do next. I'm really excited to hear that kind of thing from this audience. So the first quick little vignette I want to show you is, we worried about the length of the polymers that the coacervates are made of. We know longer polymers make coacervates more easily, make more stable coacervates, make coacervates that the inside composition is a more different solvent environment than the outside. And so you have to ask yourself, is it okay that they have to be so long, right? Do they have to be so long? And there was evidence in literature, especially from Stephen Mann's lab in Bristol, saying some years ago that no, they don't have to be long. You can still make coacervates out of pretty short things. And so that was encouraging. But the open question was, so if you make coacervates out of these short things, would they work as compartments? Could you still use them? Could RNA still go inside? Would it concentrate as well? Would you still be able to control what it did in there? How would the compartment properties vary compared to the same kind of compartment made out of a longer polymer? And so we've tried to ask that question, and the work was done by Fatma Pirchekmek and Seyhan Choi. Fatma was a graduate student in the lab at the time, and Seyhan still is. And what they did is designed a really simple system where we're using homopeptides, so polyaspartic acid, lysine, so not particularly prebiotically relevant, but a simple enough system to hopefully interpret. And they did a lot of experiments like this. We also used some other homopeptides, but I'm going to spare you that detail. And we looked at, as we increased the length of each of them, the cationic and anionic species, everything that's a red dot here made coacervates, and the unfilled spots did not make coacervates under our conditions, which were not very salty. So we were like trying to help them phase separate by not putting in too much salt. So more of a pond than an ocean, if you will. And then we selected some with increasing multivalency to compare and to ask how they performed as compartments. And what we found is, sort of counterintuitively, the compartments made of smaller molecules were better in certain ways that matter for prebiotic encapsulation. So in this center panel, what I'm showing is the concentration of single-stranded RNA. It's a 20 mer. And I don't remember the sequence, but it was something designed not to fold on itself. And what we're finding is if we put the same amount in solution, and then we look to see how the concentration inside compares, these are the shortest peptides that we made compartments from, so a 10 mer and a 5 mer together, all the way up to 100 mer and 100 mer, which is sort of more typical in literature for studying coacervates. And you can see it's very sensitive to the length of those polymers. And in fact, you do much better in terms of encapsulating that RNA. when the coacervate components are shorter. It's not, although it's counterintuitive because the inside is more similar to the liquid in these cases, the mechanism for RNA partitioning is based on ion pairing, and it's easier to kick out an aspartic acid, which is the competing anion that's only 5 long, than that's 30 long if you're a 20 mer of RNA. And so it makes sense that it would work this way. We then looked at what happened to the duplex stability of 2 RNAs that were stuck together via base pairing. And so this, I think they were 10 mers. I didn't put it in the slide, I'm sorry. It's in the paper though. And we're comparing whether they're more or less in their duplex state than they would have been in buffer. So these 2 complementary strands, this is how much of the fluorescence resonance energy transfer. Don't worry if you don't know what that is. It's just, are they close together, the 2 strands? And if they're bound together, they'll be close together. And this is what we'd expect for sort of, we're bound together as much as in buffer. And then if they're not, if they're non-complementary or otherwise have melted apart, this is what we'd expect. So if we then compare those buffer values for the 2 extremes to what happens inside the coacervates, the shorter ones were like buffer, they stay together as a duplex. The longer ones, they start to melt apart. And this destabilization of the duplex is something that's been seen for proteins that form phases inside of your cells and my cells. And the people who discovered that, they thought, oh, that's pretty cool. It's sort of a primitive kind of helicase activity that you could have without any real enzymatic activity. And so should we be sad that we don't have that with these short ones? I don't know, maybe we should be happy. It sort of depends how far back you want to think in terms of how long would these molecules have been. I would hypothesize that if you have relatively short nucleic acids, it's probably better not to destabilize their sort of nascent connections. But as they get longer, it might be more beneficial to be able to destabilize them a little bit, which could be very convenient in that as they're getting longer, the molecules that can make the compartments would also be getting longer. So the second little quick thing I wanted to tell you about is about trying to couple in some wet-dry cycling. And we just heard some really inspiring work about wet-dry cycling in the last talk. We were wondering if we had some sort of warm little pool and there were coacervates in it, what would happen if it underwent wet-dry cycles? And we did dry all the way to dryness and rehydrate, but those experiments are hard to do quantification on in the dry state. And so what we did instead is we would stop at like 10% of the original volume so that we could measure all the components and what was going on inside. Because here we're not trying to oligomerize anything. We just want to know what happened to the compartment function. Is the RNA still inside? Did it come outside? Did it transfer between them? And so to sort of try to make a long story short here, Dr. Hadi Faraz did this work, and he started by making sort of detailed phase diagrams, which I'm not going to go through slowly, but they were really important to this work. He used for this some synthetic polymers because they're cheap and you could make everything in large volume to really get all the quantification that we wanted, but it's still based on ion pairing. And he found conditions where he could start in his wet state with coacervate droplets, and he could end once he concentrated everything to 10-fold more concentrated by drying. He could end with still having coacervates, or he could start with coacervates and not have any coacervates at the end, or he could start with just barely having coacervates and then make the coacervates sort of more prevalent at the end. And this all depended on sort of where he started, like what was the composition in that little pond to start with? Where was he on the phase diagram? Because when you concentrate, you're increasing the concentration of both the polymers, which should make you phase separate better, and the salt, which should make you phase separate worse. And so it's the interplace between those two that dictates what you're going to actually have. So then we asked, okay, so for the ones where there's a compartment the whole time, what's happening to a molecule that might have been inside? Because it's kind of obvious if the compartment goes away, the molecule's got to be released, right? Or if the compartment forms, okay, the molecule is going to be taken up. But what if the compartment is always there? And the answer was kind of interesting. Because of these sort of competing interactions between sort of driving compartmentalization by increasing polymer concentration, but also increasing the salt, which acts against that, what we found is that if we go from, so this is time 0 and time 160 minutes, so this is about 10-fold concentrated compared to that. In the images, the red here is a fluorescent 15 mer of uridylic acid, so an RNA, and looks about the same, right? If you look at the quantification here, the U15 concentration, it stays between roughly 10 and 15 micromolar inside the entire time. And it does that despite the fact that the overall amount in the container goes from 0.02 micromolar to 0.2 micromolar because of the 10-fold concentration. So what's happening? Well, what's happening is the extra RNA, if you will, is ending up mostly outside. And this is the partitioning ratio inside to outside. It's dropping like a stone as we concentrate the system. And this can be understood, again, from the phase diagram because of the effect of that increasing salt destabilizing the structures, which is also giving rise to their apparent increase in volume that you see here. We can sort of make a bigger version so you can measure that increase in volume, and it's real. And so even when the compartment does not itself come and go during the wet dry cycle, you can still have the RNA becoming more mobile and then again less mobile. We watched also the diffusion coefficients of these as a function of the drying, but I'm not going to bore you with that. But it's as you would expect, right? When more of it can get out, it's easier for it to exchange between droplets. And so during the dry cycles, the system behaves, if you will, almost wetter. If the water content relative to the outside phase of the inside phase becomes larger as the pool would dry. So that to us was pretty interesting, and it's quite counterintuitive and could be important for how things would change during a wet dry cycle that involve these kinds of compartments. So the last little thing I want to tell you about is something that we just published a couple months ago. I love coacervates, and everything has been about coacervates. So we're going to make the protocytoplasm. We love a coacervate. But the truth is I also love a vesicle. We've seen some talks about vesicles, and you can see why people love vesicles. Lipid self-assembly is fantastic. It's a great way to compartmentalize things. It wants to happen on its own, right? Dried lipid films want to form vesicles. All of the cell membranes that we have in biology today have lipid bilayers, and so almost all anyway. And so we really would like to have some kind of way of forming a lipid bilayer, and so we'd like to have this cytoplasm ultimately become the core of our protocell. And presumably we'll go through some sort of incomplete amphibole layer in between, but we were curious about what might happen along this pathway. And we're not the only people who've ever been curious about this. But just to sort of catch you up to speed with a quick background of some of the things people have tried. In my lab, we kept trying to add vesicles that we had already made. So like 100 nanometer little vesicles. We would add them to a thing like this. They would stick on the outside. They made nice pictures. The pictures were very uniform and beautiful, but it had essentially no barrier function. So it wasn't helping to sort of define what stayed in and out, right? The coacervate was defining what stayed in and out based on equilibrium partitioning. And the membrane, if you will, was just a bunch of liposomes attached there, and it didn't gate entry at all. It stopped coalescence, but it didn't gate entry at all. Some really nice work has come out of the Mann lab, again at Bristol, where they've put in molecules that weren't pre-assembled. So either below their CMC or in a solvent like ethanol. And those also assemble around the outside of a coacervate droplet. And they form, again, things that looks relatively uniform in a microscope, but they have some kind of perm selectivity control that they offer. And especially for the ones in this 2021 JAX paper, they let little molecules get in, but higher molecular weight molecules are kept out, which is a pretty useful thing, both for artificial cells, but potentially for a protocell that wants to be able to have food come in and waste go out without having to evolve complicated membrane proteins. In all cases, again, like all the structures within a system look kind of similar. And we were curious what would happen if you tried something like a dry lipid film, because that's the kind of lipid self-assembly that we're already imagining is happening in these sort of prebiotic environments. So instead of bringing the lipids in either as a preformed vesicle or as individuals, what happens if they just come in as a dried lipid film? So this work was done by Ali Marinelli and Fatma again, both as graduate students in my lab. They've graduated recently. And we're comparing now just the gentle hydration, which we've heard some about already. We didn't do anything special to make the gentle hydration particularly favorable here. And you can see like it makes some big vesicles, but it makes lots of other stuff, which is typical for gentle hydration. These are phospholipids, not particularly prebiotic in terms of their composition. But yeah, hold that thought. At some point, we are starting to do it now with other mixtures of lipid. So this now is looking at the same exact experiment, but the only change is instead of just adding water as the hydrating solution, we're adding coacervate suspension. The droplets are already in there. There's sort of sediment down on the surface. You come in and look at it, and you see these beautiful things that look like vesicles, where the lipid is assembled directly around the outside of each one of the coacervate droplets. And we've spent a fair amount of time characterizing like what those structures actually are, and we don't have time to go through all of it. But I will say two things about it, if Uli lets me. I don't know what time I'm supposed to stop talking. I could skip this one if I had to and go to the next one.
Soon. You want to wrap up soon within the
Okay,
next
so they're coacervates, so they get stuff inside really well. Ones that don't have coacervates by gentle hydration do not. Okay, so, but if you don't have the molecule there before the lipid membrane forms, if you only bring in a solute after the lipid membrane forms, you can ask the question about uptake, right? How intact is the behavior of that lipid membrane? And these experiments were done with different coacervate systems. It's not particularly important what they are. Neither of them is super relevant to prebiotic chemistry, although they're different enough that probably this is going to carry over to other systems. And what we looked at is two different fluorescent solutes. The one is this 15-mer of uridylic acid again, and the other is a small molecule dye, because we wanted to provide a sort of harder test for the membrane. And we put it outside and we come back and take some pictures 10 minutes later just to see if those molecules got inside. We know the molecules at equilibrium want very much to be inside. And you can see the ones, the droplets that do have molecules inside have quite a lot of molecule inside. But what I want you to notice is the less obvious droplets in between. These are all coacervates inside too, but they didn't get molecules inside. And you can count them all up. Better than half of the droplets resist entry of any of these molecules over the time course of this experiment, and longer. These pictures just happen to be taken at 10 minutes. Something really changes after. And the other thing that's notable about this is that, so they've kept little molecules out, which is something we hadn't seen before, either adding the lipids as pre-assembled as vesicles or as individuals. Forming from the lipid dried film actually made half of them better quality membranes, but only half of them, right? The other are worse quality membranes, and they even let in the U15. And so this variability in permeabilities from different individuals in the population, sort of as a physical chemist who often makes things that I want reproducible and to make all the same, it makes me a little sad and I want it to be more perfect. But thinking about prebiotic chemistry, if you make perfect membranes at the very beginning, well, then what can you do, right? Now you're really missing the fact that you don't have any gating to have membrane proteins to allow things in. But if you make sort of this mixture of different behaviors, there's opportunities for selection and competition in this population, and potentially, once we're housing some reactions, some kind of evolution. So we think that could be pretty interesting going forward. And you can read the take-home messages. It was a short enough talk, you probably remember them. I introduced the folks as I went through, and the money came from NASA, so thank you all for your attention.